have the exams here ready to be given out, and glad you're enthusiastic. And so we'll stop. I'll try and stop lecturing. Remind me if I don't shut up at about 12:30. No, 1:30.
That's 30.
It's good to stop at 12:30, isn't it? So we'll go away. 1:30, and I will hand out exams like we did last time. I don't know how many of the TAs are going to show up, but I'll announce the alphabetical letters that each of us is dealing with. And if you could come forward, because they are in alphabetical order, come forward if you're at the beginning, hang back if you're at the end of that. Group will help a great deal. And last time we got them all done in plenty of time, so because we have, quite a bit of time before the next class starts. So we'll do that. I haven't put up the grades online because I don't want you spending your time. I only just finished entering them about an hour ago and checking them, and I don't want you spending your time looking them up on the computer now. And ditto for the key. I'll put the key up in about 2 hours once I get back and finish it up. I have the key in written form, the one I give the TAs, but I need to put it into the computer. So if you have questions about the questions, once I've finished handing out the exam, let me know, otherwise the key will be up. Anything else? Yes. Oh, the average was a few points less than before, but it was, about 78%, something like 78% for the whole class as a whole, which is, fine. It means that at least 1 in 5, and maybe about 1 in 5 of you got, straight A's or A minuses. A pluses or A's, 4.0 GPAs. And overall, since you did somewhat better in the first exam, I think it's about 1 in 4 have reached that level of attainment. So, so I'm not planning on making any sort of major changes in terms of extra credit or anything like that in the final. I will send out bunches of information about the final. There's a couple of things I do for every class, which those of you who are in 330 with me last year will know. The first is I always tack on 5 points, 3, on top of the final score, and that's to compensate you for the fact that most of you, Christmas is coming up, holidays are coming up, don't want to argue about regrades, and so I just give everybody 5 points, which you can use towards that total that you need for a given grade. Okay, so if you don't want the 5 points, you can, or you feel your exam was, I usually hold a morgue where people can come and look at their exams after the test, and so you can always come and try and get more than 5 points, if you wish, if you think they're there. But believe me, I think it's rare. It's only a 125-point exam. So if you're trying to predict your grade, you can add that 5 points onto the final straight away. And the other thing is, should you do exceptionally well in the final, in other words, should you get an A in the final, but a B plus overall, I will give you the A. So essentially, I will give you the better of the two, either your overall grade or the percentage in the final. So you can go online and figure out from the grade. You figure out from the grade the list of points. You divide them by 275. You can figure out what the percentages are that you need. And so if you get, say, if overall in the course you need an 88.5 or something to get an A in the class, or whatever it is, or A minus in the class, then if you get that in the final or more, I'll give you that grade. Is that clear? Yeah. If you do worse in the final, I don't care. I don't want to know about it. Okay, but there are very few of you, almost none of you, who can afford to blow off the final. So don't even think about it. It's just that I recognize that sometimes it takes a couple of goes rounds with these complex issues before the light bulb goes off. And don't, you've taken the second exam, so you don't have any choice now, but don't plan for that, because it is actually quite difficult to do better. Just because the final has more material in it and you have other final exams, it's quite difficult to do better in the final than your overall grade. It's easy for me to say, it's an encouraging thing, but the number of people who I award increased grades to is, a smattering of people through the class. So don't kid yourself about it, but it's something, especially helps with people who are struggling in the class, who, really put it together for the final. That's actually who it's mostly aimed at. Okay. I'll also I'll send you an email about the final, about the topics that will be covered. In general, if the broad topic isn't covered in the previous 2 exams, this is for the cumulative part, it won't appear in the final. So for instance, we didn't cover, for people who took the main exam, we didn't cover auditory transduction in hair cells as an exam topic. And therefore that will not be on the final. Okay? Yes? So you don't have to learn all of the material. Yes? It'll be the same format, although again we need to get it graded. There's now a limit with the final. We have to get it graded within a couple of days. So that puts some constraints in terms of essays. But I would like it to be the same format. It's just it's not always possible. Yeah.
So only material from the first 2 exams specifically will be on the final?
No, if you look in the syllabus, there's a cumulative part of the final, which is worth so many points. And then there's the final, the questions about the final series of lectures.
Cover the transduction of the auditory system, so
that will not be, if it wasn't covered in the previous 2 exams. So the way to review your material for the previous 2 exams is to look through the old exams, to figure out broadly what is this question about, yeah? To make damn sure you know the right answer to the question that was presented in the previous 2 exams, and then to broaden your knowledge of that question out by a few slides in the lectures. So, for instance, there was questions in this exam on orientation selectivity in the cortex, and there was a question on ocular dominance pathways in the cortex, and so that topic is the whole topic of the projection from the eyes to the visual cortex is something I might ask questions on. Okay, because the way I write the exam is to go through the questions that I've asked in the past and say, well, how can I ask this in a different way or hit this subject in a different way? Yeah? Yep. Is that fair enough? Okay. And I deliberately don't write a list of topics that you have to learn and topics that you don't have to learn, because I would love to trick people who are really paranoid into learning it all. That's a wonderful thing, and I'm surely not going to stand in the way of that. And secondly, I think that knowing what a question, what subject, broad subject a question is asking about, is something that you should be able to do, part of the training. So, okay, it's worked out well in the past, and if you want to know whether a particular subject is going to be mentioned, can't figure it out, you're welcome to use the discussion board. Yeah? Okay, so I will write out an email with all of that stuff in it once I have the key and the exam results up on Blackboard. That will be my next task, the next thing you'll be hearing from me. Okay. Okay, so this is the an image. Let's get the lights down a little here. Okay, this is, as I mentioned before, this is a section through your skull showing the position of the olfactory epithelium up there and the olfactory receptors and the odorants coming in here. And we talked about the olfactory epithelium, where you have the purple olfactory receptor cells, the grey olfactory receptor cells with the purple nuclei here, and how they have cilia projecting out from this sort of knob here, dendritic knob it's called, and these cilia contain the transduction apparatus, and that transduction consists of the odorants initiating or eliciting a flow of current of positive ions, sodium and calcium, as usual, into the cilia, through the plasma membrane, into the cell. And then that flow of sodium and calcium depolarizes the olfactory receptor. And then the olfactory receptor up here has voltage-gated sodium and potassium channels, which will detect that depolarization. And when it reaches threshold, they'll initiate action potentials, which then travel up through the cribriform plate and up into the olfactory bulb. And we'll talk a bit more about what happens up there in a few slides. And this flow of inward current is known as a generator potential. That's sort of the generic term for a potential that's initiated by stimulus in that sensory neuron. Okay, so a simple experiment is to wholesale clamp or patch clamp, wholesale patch clamp the olfactory neurons and voltage clamp them to their resting potential, and then measure the response to odorant, which is spritzed onto either the cilia or into the cell body. And when you do that, you see this inward current that is elicited with a small, short delay, about 100 milliseconds or so, 150 milliseconds. And this inward current is the sodium and calcium current that's carried into the cilia. If you spritz deodorant onto the cell body here, you don't get much current, and after a much longer delay, indicating that it's the cilia that contain all of the transduction apparatus. And then that inward current, as I said, is converted into a string of action potentials, whose frequency codes the amount of odorant that's present. Okay, so what does this delay tell you? It's about 100 milliseconds. Would that indicate, because there's a couple of ways that this could happen, right? Well, what are the ways in which an odorant could stimulate an inward current? How could that work? Yeah?
Via metabotropic receptor?
Could be through a metabotropic receptor, or it could be ionotropic receptor, okay? Because in this sense, the odorant you can think of as analogous in a way to a neurotransmitter. Obviously, it's a very different sort of evolutionary. The significance is very different for the organism, the evolutionary impact of it. But nevertheless, sort of mechanically, it's just a small molecule that binds to the cell membrane, something on the cell membrane, and initiates current. So given that it takes about 100 milliseconds for the odorant to have an effect, what would you guess? Is this metabotropic or a channel? Metabotropic, right, yes. Okay, and so let's look at that. And the other interesting thing here is the cilia, because we mentioned that the outer segment of rods and cones is also a modified cilium, a single modified cilium. And so there's some questions there about whether this sort of similar morphology or ultrastructure of the olfactory cell or betrays a sort of a kinship in terms of mechanism of transduction. And we'll see that's true as well in a way. So are cilia involved in the olfaction process, and does that have any significance? So this diagram shows you your the plasma membrane of the cilium. So now we've zoomed in just on a little piece of the cilium. So up here is the mucus, right, that contains, overlies the olfactory epithelium. And so up at the top is the nasal cavity. And so odorants are arriving from the top of the sheet there, from the top of the page. And they're traveling through the mucus, maybe via binding proteins, maybe not. But they're traveling through the mucus. And they interact with a receptor protein that is, surprise, surprise, a G protein-coupled receptor, right? And it interacts with a heterotrimeric G protein, 3-subunit G protein. It splits off, when it binds to the G protein, it splits off the alpha subunit with GTP bound to it instead of GDP. That's the activation process. I guess these little guys stand for the phosphates on the GTP. I don't know. So this G protein goes by the name G-olf, or golf. And G-olf is named because it's the olfactory G protein. It's a separate gene from the one that mediates vision, transducin, or the metabotropic neurotransmitter receptors. So ones that bind to metabotropic neurotransmitter receptors. And the target enzyme in this case is not phosphodiesterase, quite the reverse actually. It's an enzyme called adenylate cyclase, and many of you have met this enzyme if you've learned about how the liver functions in 330 or in mammalian physiology. Adenylate cyclase is an enzyme that converts ATP to a small messenger molecule called cyclic AMP. And so cyclic AMP then binds to a sodium-calcium channel, a rather non-selective cation channel, and lets sodium and calcium inside the cilium, and that is the cause of the inward current flow that occurs, or at least some of the inward current flow. Now, there's an additional little sort of curlicue on this, and it relates to the fact that in order to generate still more inward current flow, the calcium ions that flow in bind to a chloride channel and activate a chloride channel. And some of you might be thinking, oh gosh, that can't be right because we've been taught that chloride is inhibitory. Chloride channel is inhibitory. Remember the GABA channel is neurotransmitter gated channel is inhibitory. And we had a long thing in the lecture here talking about which way the chloride flows in most cells and determining that the chloride throw in the neurotransmitter synapses, postsynaptic membranes of synapses, chloride flows into the postsynaptic cell, and that's why it's inhibitory, right? It's a negative ion flowing into the postsynaptic cell, and so it's an inhibitory action. But in this case, the mucus outside here happens to hold a very low concentration of chloride ions. You know, it's much more like water, right? It's part of our extracellular environment, or part of the environment that's outside our body fluids. Right? We know that because it comes out of our nose, right? So the mucus and all that sort of watery fluid stuff that comes out of your nose when you sneeze or blow your nose, that material, that fluid, is quite low in chloride ions. So the chloride ions here actually flow out when the chloride channel is gated by calcium. This calcium-activated chloride channel, it opens. Chloride ions flow out because there's much less chloride outside than there is inside. And this flux of chloride out here constitutes an inward current, right? It's a negatively charged ion moving out of the cell, and that's equivalent to a flow of electrical current into the cell. Put another way, when these chloride ions leave, they're going to leave counter ions inside, positively charged counter ions inside, and those are going to result in a net positive charge being created. And so the flow of inward current and the consequent depolarization of the olfactory receptor cell is amplified or enhanced by the action of this calcium-activated chloride channel. Right? And so you might say, well, isn't that a small little curlicue? Why do we have to learn about that? It's because calcium-activated channels are a whole big family of channels. And it's nice, and so it's good that at some stage in this course we come across their use. And it's a really neat example of evolution adding further steps to an already complicated story in order to generate, in this case, more amplification. Now, out here is a sodium-calcium exchange protein because obviously if olfactants are present, a lot of calcium is going to enter the cell. And so that calcium has to be removed, and the calcium is removed by this sodium-calcium exchange. And the other thing to say about this is that the calcium that enters here will bind to proteins like calmodulin, and that will do all sorts of things inside the olfactory cell in terms of long-term changes of function. Okay, so essentially it's a G protein link cascade mediated by adenylate cyclase and cyclic AMP, and the essential first step is the activation of a cyclic AMP-gated sodium and calcium channel. Now you can compare this with the rod. Has the rod got a G protein linked receptor, the rhodocone? Has it got one of these guys? G protein linked receptor? Good. That's rhodopsin, right? What's the difference here between in the rod and cone? What's the difference? Yeah. Oh, yeah. Sure. Yes, Steve.
That's phosphodiesterase.
Right. So it's actually kind of the opposite, right? The rod chews up cyclic GMP in the light, whereas the olfactory neuron produces cyclic AMP. I mean, it's not exactly the opposite, but it's cyclic AMP and cyclic GMP are closely related compounds. And then, what about this guy here? So is there one of these in the rod? A cyclic nucleotide gated channel? There is, yes, just cyclic AMP instead of cyclic GMP. Yeah? It is a surprising thing about the eye that it's not faster, right? And it's, as I mentioned, it's the thing that makes the movie industry work. I think the answer to the question is that both the nose and the eye require extraordinarily high levels of amplification. So in dim light, the eye, photoreceptors in the eye of almost all organisms are capable of generating large electrical responses to single photons of light. Yeah? And in the olfactory epithelium, there's been some controversy about this, but I think the net conclusion of the work that's been done is that these olfactory receptors can produce detectable electrical responses. That's little blips of inward current in response to the binding of a single olfactory olfact molecule. Did I just run out of power? Let's see what I can do. Otherwise, it's going to be back to shouting screen. Yeah. There's something here. Okay. Better? Tell me if it goes again. Okay, yeah. So I think the thing is that the need for my view is the need for amplification overrides the issue of speed in both cases. Cones are a little faster; they have less amplification. But I think by the time cones, the rods and cones sort of diverged, the pathway for visual transduction was already set. So the chances of, but essentially, if visual transduction was mediated by a rhodopsin that was a channel, right? So that the photon hit the rhodopsin molecule and it became a channel, so it was more like an inotropic receptor. If it worked like that, it would be extraordinarily insensitive. We'd only be able to see under quite bright light, would be my prediction. Because the activation of a single channel just does not pass very much current. Okay. Yeah. Okay, so here's the G protein linked receptor, 7-transmembrane domain, the odorant receptor, and as I mentioned before, this family is a huge family of related G protein linked receptor proteins, olfactive receptor proteins in your genome. And I did some research just the other day, and I think I gave a number of 500 distinct genes in humans that code for odorant receptors. That number is up to about 1,000 now. So there are 1,000 different versions of this basic protein sequence, each with a slightly different primary sequence. And that is about 3 to 5% of all our genes. And we're not exactly olfactorily motivated animals. Something like a bear or a dog probably has a higher percentage than that. Yes?
How can you explain like carbon monoxide that's like odorless? Like how does that work?
Carbon monoxide?
Right. Isn't that odorless? Like I'm saying like how...
Oh, how it's odorless?
Yeah, like, you know, like does it just not bind to the...
Yeah, I mean, if you look at the... I don't have the slide here, but if you go back to the last lecture and look at the molecules that are odorants, they're all fairly substantial molecules. Alcohol is one of the smallest that is an odorant. So a molecule like carbon monoxide is probably not going to bind to any of the pockets of the odorant receptors. That's my guess on that. Kind of unfortunate, though, right? If it were the case, we could, like natural gas, but, you know, natural gas we can smell, and we still blow ourselves up with it quite regularly. I'm not sure that evolution would really favor our ability to detect carbon monoxide. Yes?
Can several different odorant molecules stimulate one receptor protein?
Yeah, let's talk about that. The relationship between the olfactory receptor protein and the odorant molecules, the sort of the spectrum of odorant molecules that will excite that single protein. So we're going to do that right now, I hope. Let's see where we go with this. Yes. So given all these olfactory receptor proteins and the fact that there are, you know, hundreds of thousands, if not millions, of olfactory receptor neurons in our olfactory epithelium, sorting this out, either from the perspective of a neuroscientist or from the perspective of our own poor adult brain, would be a nightmare if there were not some simplifying assumptions. You know, the same kind of nightmare as I talked about that would exist if Dale's principle wasn't by and large true. And so the principle that turns out to be important is that each olfactory receptor neuron expresses only one, or at most a few, odorant receptor proteins. And let's just say, for the sake of argument, just one. Okay, it's rather like Dale's principle. Of course, there are exceptions. Okay, so that simplifies things a lot, right? And of course, there's a big question there, which is how does a particular, how does that happen during development, during the creation of olfactory receptor neurons? But we're not going to tackle that here. Now, that means that olfactory receptor neurons expressing the same receptor protein are scattered across a wide region of the olfactory epithelium. So it's not as if all the olfactory neurons, receptor neurons that express one kind of receptor, one of these 1,000 genes, are concentrated in one part of the olfactory epithelium. In fact, they're spread, scattered throughout it. And so the olfactory epithelium therefore consists of a patchwork of neurons expressing different olfactory receptor proteins. Right? Any one neuron only expresses one, but in any one region of the olfactory epithelium, you could find dozens of different types of olfactory receptor neurons which are expressing each an individual receptor protein. So we thought we were making life simple here, and then suddenly it becomes dramatically more complicated because we can't cluster these families of receptor neurons into discrete parts of the olfactory epithelium. Okay. So now we get into the fine print here. And the first thing to consider is you know, does it matter that the olfactory receptor proteins that express a given odorant are spread out? How, in fact, do we distinguish one odorant molecule from another? What do we need to do that? And there's a very simple sort of principle here that's not easy to grasp sometimes, but it's real simple. And it goes like this. Okay. So each odorant receptor protein is quite broadly tuned. In other words, again, it would be really nice and simple if each odorant receptor protein only recognized one odorant molecule. That would be really nice. But it's not like that. Right? A given odorant receptor protein is capable of recognizing dozens of different odorants with quite different chemical structures. Okay, and Ricardo Araneda spent quite a bit of his life as a postdoc trying to sort of characterize these sort of spectra of odorants that would activate a given olfactory receptor protein. So that's the first sentence. Each odorant receptor protein, and therefore receptor neuron, right, is quite broadly tuned, responding with varying sensitivity to a range of odorant molecules, not just exclusively to a single odorant molecule. And so, again, life just gets that much more complicated to figure out how this actually is working. So a consequence of that first statement is that if our brain was to measure the response of only a single odorant receptor neuron, it would not be possible for our brain to determine whether the receptor was responding to a low concentration of one odorant to which it was highly sensitive, or a high concentration of another odorant to which it was relatively insensitive. So it's not possible for the brain to just look at the action potential frequency coming from a single odorant receptor and figure out what the odorant is that's out there. Yeah? What the brain has to do is compare the activity of one odorant receptor, which is tuned to one set of molecules, with another odorant receptor that is tuned to an overlapping set of molecules. Right? And it's only by comparing the output of a number of different olfactory receptor neurons that the brain can figure out what olfactant is there. Yes?
So is that sort of like, the colors?
Exactly. It's exactly the same issue that we face with color vision. You can't, although we have these things called red cones and green cones and blue cones, right? If you only had red cones, you couldn't distinguish one color from another, right? Because you couldn't tell whether what you were looking at was a dim red object, right, or a bright blue object. They would both stimulate the red cone by about the same amount. Yeah, the red cone's not very sensitive to the blue, but it's bright. Okay, so that's a way to, perhaps some of you, it's easier to think about that way. So yeah, so in order to measure color, we have to compare, the brain has to compare, or the retina has to compare the output of all three cone types, right, because they all respond to overlapping wavelengths. Yeah?
So the odorant receptor proteins are all kind of like the vision ones, in the sense that they sense it and they need each other to really distinguish and define the different odors?
Yeah, I mean, so let's look at vision. I'm going to spend a bit of time on this because it's important to me. So let's look at vision, right? Let's go back and think about color vision. So if this is wavelength, and this is absorption by the pigment, by rhodopsin. Right? You've got your three cone families here, right? You've got your short wavelength, or otherwise known as blue, and then the medium wavelength, and the long wavelength, right? Okay, so let's think of a wavelength. Let's think of two wavelengths, one here and one here. We'll call these wavelength 1 and wavelength 2. Now, if you only had your short wavelength cones, if you only had one kind of cone, you couldn't distinguish between these two wavelengths as having distinctly different colors. Right? Why is that? Because wavelength 1, admittedly, is easily absorbed. Right? And wavelength 2 is less well absorbed. Yeah? I grant you that. But you can't use that to distinguish between the two as being different. Because you could be looking at a high intensity of wavelength 1. Sorry, you could be looking at a low intensity of wavelength 1 and a high intensity of wavelength 2. Yeah? And in that case, the absorption would be, the total absorption would be the same. Yeah? Because the short wavelength cone is relatively sensitive to wavelength 1. It's relatively insensitive to wavelength 2. So if you have a lot of this guy and not much of that, you couldn't tell the difference, because all you've got to use to tell the difference is the hyperpolarization of the cone, or the action potentials coming from the ganglion cell that's attached to it. And that doesn't tell you what the color is. That just tells you how much net absorption of light there has been. And in the case that I'm mentioning, where you have a low intensity of this and a high intensity of that, then you can't tell the difference. Yeah? Now, if, though, you have both short and medium cone information available to you, then let's look at a wavelength like this. Then no matter what the intensity of light at this wavelength, no matter what the intensity is, you will see a high output, a high response from the short wavelength cone, and very little response for the medium wavelength cone, no matter what the intensity of this light is. Right? Because at every intensity, the medium wavelength cone is going to absorb less light. And so by comparing the response of this one and this population of cones, the brain can figure out, you can figure out, that this must be a wavelength here. Okay, if there's a wavelength here, we'd have more from the M. If it was the wavelength of the stimulus was here, we'd get equal signals from our S and M cones. Yeah? Yes?
Are cones in your fovea, like, patchy,
Yeah, you've seen the picture. The cones in the fovea are a mosaic of red, green, and blue cones, of short, medium wave cones, yes. Because you sort of want them randomly distributed, right? Yes?
So if we only had one cone, would we just see in black and white?
If you had, this business of seeing it in black and white is not the way to say it, right? The way to say it is, could we tell one color from another? And if we only have red cones, we can't. Yeah? I mean, the chances are that red objects, if we only had red cones, the chances are that red objects would be brighter. Would appear brighter to us, yes? But that's assuming that all the objects out there are emitting the same amount of light. And then I agree, the red ones would appear brighter. Right? But that's not the way the real world is. And if we only had red cones, we couldn't tell the difference between a dim red stimulus and a very bright blue stimulus. They both look like shades of grey, equivalent of shades of grey. The same shade. Yeah? Okay, so the olfactory system, in order to decipher which odorant is present, go back to the olfactory system, the brain must compare the output of an array of different odorant receptor neurons. Right? So for the olfactory system, this axis would represent different olfactants. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. These would be olfactant structures. Olfactant molecules, right? We just labeled them arbitrarily 1 to 10. Different kinds of structures of olfactants. And any one of the olfactory receptor neurons would be sensitive to a range of these olfactant molecules. And again, by comparing the output of, say, receptor neurons that express this receptor with receptor neurons that express this receptor, the brain can determine what olfactant is present in this range here. Yeah? Yeah? Okay, if olfactant with the chemical structure number 2 is present, there's going to be a lot of activity from this receptor neuron and almost none from that one. If number 4 is present, they're going to be equally active. Right? And that determination is not going to be dependent upon the concentration of the olfactant, because it's a relative measure. Okay. But of course, the amazing thing about the olfactory system is that there are hundreds of overlapping families of genes, and there are hundreds and thousands, maybe thousands of molecules that we can actually discriminate with our nose. Right? So it's the visual system on steroids. But the visual system has a really simple problem to solve, to determine wavelength. Olfactory system has a hugely complex computational problem. And so what the olfactory system has to do is it must compare the output of an array of different olfactory receptor neurons. So it has to compare the output of this one, and 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 this one. Okay? And compare and label a specific pattern of relative activity amongst these populations as indicating a specific odorant. Right? So, in other words, the brain has to label, if these guys are all active but these ones are not, well, the odorant is number 6 to 10 or something like that. Yeah? Okay, that's what the task the brain has to do. So presumably stimulation with a particular odorant creates a particular pattern of activity across the olfactory epithelium, which is then mapped onto neurons in the olfactory bulb. And that's a kind of topographical mapping process. Right? Now that mapping is where life gets simple again. And the light bulbs will go off when you consider this mapping process. So here's the olfactory receptor neurons down here. And they send axons that project through the cribriform plate here to these these sort of tangles of synapses. And these tangles of synapses are called glomeruli. You've come across glomeruli in the kidneys. Well, these aren't they. These are other kinds of glomeruli. And as you can see here, in each glomerulus, synaptic contact is made between a group of receptor neurons, 2 in this case, and one or more of the second-order cells. And those cells are called mitral cells. These ones here, these are called the mitral cells. Okay, here's a mitral cell. Okay. And so, in the mouse, for instance, approximately 25,000 olfactory receptor neurons in a small part of the epithelium converge onto 25 mitral cells in each glomerulus. So here we've just got 2 converging. You've got to realize that, especially in animals that are very sensitive to odors, there may be thousands of olfactory receptor neurons converging onto the synapse, making synaptic connection with a single mitral cell. And of course, the function of this convergence is to provide a huge amount of amplification, so that the excitatory synaptic potentials that are generated by each olfactory receptor are summate to provide a very substantial signal to the glomerulus. And then there are granule cells, which are interneurons here as well, which perform the task of comparing what's happening in one mitral cell to another, rather like the horizontal cells and the amacrine cells in the retinal system. So these mitral cells have axons. They produce action potentials. They have axons that go up into higher levels of the brain. Okay, so where does life get simple? Well, life suddenly gets simple when you consider the nature of this connection here at the molecular level. It turns out that all of the olfactory receptor neurons that converge onto one glomerulus all express the same receptor protein. Right? So every olfactory receptor neuron that converges onto, say, this glomerulus will express one particular olfactory receptor neuron protein. And all of the olfactory neuron axons that converge onto this glomerulus will express a different, will come from olfactory neurons that express a different receptor protein. And so the array here of glomeruli in the optic lobe, sorry, not the optic lobe, the olfactory lobe, the array of glomeruli in the olfactory lobe constitute an array of output from individual receptor proteins. Yeah, because activity in this glomerulus would reflect activity from these two neurons, and both of these are expressing the same receptor protein. Activity in this glomerulus would reflect activity in these two, and these two are expressing a different kind of receptor protein. And so this sort of patchwork of olfactory neurons in the epithelium, each of which expresses a different receptor protein, a sort of crazy quilt, through this convergence simplifies into an array of a much smaller number of glomeruli, each of which responds as a result through the activity of an individual receptor protein. And so activity in this glomerulus would reflect, so that this glomerulus here might respond to olfactant structures in this range here, 1 to 5, and activity in this glomerulus here would reflect activity of a different set of olfactant molecules. And by comparing the activity in this glomerulus with that glomerulus, i.e. the activity of this mitral cell versus that mitral cell, the brain can begin to determine what kind of olfactant is present. Yes?
So you have maybe thousands of olfactory cells, and then few, like they all converge into one glomerulus?
Yes.
And then from the glomerulus they converge into even less?
No, we'll see where the axons of the mitral cells go to. They actually branch to a number of brain structures. And there's not that much understanding yet of what happens at that level.
Okay.
Yeah, so but there must, I guess there must be some convergence of information higher up, because you have to compare the activity of one mitral cell to the next. So there has to be some convergence, right? Just as there has to be convergence in the color system, and that occurs in these retinal ganglion cells that you mentioned, where you have centers and surrounds with different sensitivities to color. Okay, so the proof of this business about 25,000 neurons converging on one mitral cell, simply because they all express the same receptor protein. Think about the cell biology of that. It's pretty amazing. It takes some convincing. But it does happen, and the proof of it is to label cells which express, to label olfactory receptors which express a given receptor protein by labeling the messenger RNA for that protein. Just doing a standard in situ hybridization of the RNA. And when you do that, you can see here, these are two glomeruli that that are both connected to olfactory receptors, which converge on them, and they're both labeled by, in this case, by through the mRNA for the olfactory receptor protein. And I think the current concept, you say, well, how can this happen? You know, how do these neurons, these olfactory receptors, find their way to converge onto the same glomerulus? How does that happen? And one possibility is that a small fraction of the olfactory receptor protein is expressed actually not just in the cilia, but also in the cell body and in the axon and the nerve terminal of each olfactory receptor neuron. And maybe that's providing some recognition between the neurons that express the same receptor protein. Okay, so each glomerulus transmits the combined response of thousands of olfactory receptor neurons, all of which express the same olfactory receptor protein and are responsive to the same subset of odorants. And so the pattern of active glomeruli corresponds to the pattern of receptor proteins activated by a given odorant. And this now, we can come back now and look at an experiment that I showed you at the very beginning of the year, where somebody's using calcium-sensitive dyes to look at the activity of neurons in the olfactory bulb. And so here is one of these glomeruli, and each of these little spots here of darkness is the fluorescence from one of those glomeruli. And the darker the spot, the higher the calcium concentration. It's sort of a reverse image. The spots, the glomeruli actually glow brighter when the calcium dye fluoresces, but it's shown as a dark spot here. Right? And then this just sort of tries to look at these. Each of these circles are glomeruli, and then the dark ones are the ones that are active. And so here's one kind of olfactant, a pineapple smell, which is presented to the mouse while a microscope is imaging its olfactory bulb. The cranium of the mouse has been opened up. This is an invasive process. The microscope's looking at the olfactory bulb here, and the pineapple is presented, and these glomeruli become active. And then when a banana is presented, a separate but overlapping subset of the glomeruli become active. Okay, and so each glomerulus represents the activity coming from hundreds or thousands of olfactory receptor neurons, which respond to a subset of the odorants that come from a pineapple. And this one here, these ones here are responding to a subset of the odorants present in a banana. Okay, so ethyl butyrate and amyl acetate are the two pineapple-y and banana-y smell compounds that are used, olfactants that are used in this case. Okay, so the fact that, for instance, trying to look here, yeah, the fact that this one here, this glomerulus here, is active to both the pineapple and the banana smell, to both the ethyl butyrate and the amyl acetate smells, indicates that there's an overlap between the sensitivities of the olfactory receptor proteins in that case. Whereas the fact that this one, say, up here, is only sensitive to the banana, but not smell, but not to the, only sensitive to the pineapple smell, but not to the banana cell, that indicates that there's no overlap. Okay, so that's like looking at two of these curves. This will be the first two receptor types there, sort of spectrum, and then the second one, or the third one out there. Third one will be out there. Okay, so these don't have any overlap, whereas the other ones do. Okay. Questions about all of that? So what happens to the information from the mitral cells? Well, it goes further up into the brain. So here's the olfactory bulb, which is perched above the nose here. we're looking sort of from below. So this would be the front of the brain here. I'm sort of looking from below. Here's the olfactory bulb. The axons of the mitral cells project into the brain here, and they terminate, they branch and they terminate in a number of areas of the cortex. So there is a direct connection between the olfactory bulb and the cortex. And this is very different from the situation in any of the other sensory systems that we've looked at, where there are many way stations on the way, the thalamus, for instance. And the areas of the cortex that the olfactory bulb neurons project to are evolutionarily quite ancient. They're areas on the rim of the cortex, the sort of lower, the undersurface of the cortex, the rim of the undersurface of the cortex. And the amygdala, which is very active during emotional events in our lives, the piriform cortex and the entorhinal cortex. So here we go from the olfactory receptors, through the olfactory nerve, through the cribriform plate, to the olfactory bulb, and then to these, directly to these targets in the cortex, these more ancient cortical areas. And then through to directly from there through to the hippocampus, the hypothalamus, the thalamus, and the frontal cortex, parts of the frontal cortex. And so this structure, this sort of very tight connection between the olfactory bulb and some fairly ancient parts of the cortex that are involved in fear here, and emotion, in memory, and in the regulation of hormonal levels by the nervous system, has often led to speculation that, well, this is indicative of the fact that of all our senses, smell has perhaps the greatest sort of grip on our emotions. And whether you like to imagine that's the case or not, maybe it doesn't have a great grip on your emotions, but that is the sort of meme or the legend that people like to spin, the tale that people like to spin about these connections and how different they are from the visual or auditory connections, or mechanosensory connections. Yeah.
Where is the glomeruli located on that diagram?
In here, it's in the olfactory bulb. Yeah, so this thing called the olfactory nerve, maybe that's what's confusing you a bit. This thing that's called the olfactory nerve are these sort of streams of individual axons that go between the olfactory epithelium and the olfactory bulb. It's not like a long nerve. Yeah, is that what part of the problem was? Yeah. Okay, so the olfactory bulb is the structure just above the, and you can see from its shape there, it's got that sort of slightly curved shape. You can see how it's this sort of bulbous structure here, slightly curved structure on the end. I think got the same colour. Yeah. And the same colour there. Okay. All right. It is, but I don't see my... Well, do I see Ron Kang? Are you here, Ron Kang? Oh, he is there. He's just hiding. I'm sorry. Ron Kang, you should have, waved or something, yeah. You almost caused us all to be late. Come on.